Hey, you guys, good morning. I'm sorry I'm gone again. I have a pulmonologist appointment. I tried really hard to get it changed, but they would not be able to see me until like February, which is not a good thing since they were trying to get rid of my cough. So I'm sorry. Your job is to watch this lecture video and then to watch the video that I have posted on the Google Stream. To then finish your lab, the diffusion lab. And if you look on the countertops, you'll be able to find your beakers. So finish the post-lab questions, and you'll have one other book assignment to work on. And I think that's it. And the assignment that was due today, we will grade tomorrow. Again, sorry I'm gone. Okay, so today I want to talk a little bit about active transport. First of all, big difference between active and passive transport is the cell has to exert energy in the form of ATP, which is a molecule that stores energy. So the cell uses energy from ATP to move molecules against the concentration gradient. It moves molecules from an area of low concentration to high concentration, which is completely opposite of passive transport. I always think of active transport like this person that's kayaking. If you notice, they are kayaking upstream. They are going against the current. So this person is going to have to do a lot of paddling and expend a lot of energy to get from point A to point B. There are three types of active transport I'm going to talk about. First type is protein pump, then endocytosis, and then exocytosis. Let's start with protein pump. So protein pumps are very similar to protein channels. They just do a little bit more work, and they move particles from an area of low concentration to an area of high concentration, an area of low concentration to an area of high concentration. I was right. I thought I said it wrong. Anyway, so a protein pump, similar to a protein channel. However, this guy actually moves and changes shapes as it moves particles from an area of low to an area of high concentration. When this guy moves and alters its shapes, to get different particles in from one side of the cell to the other, it has to expend energy. So that ATP is just representing that this protein pump needs energy to move these particles from here to here. So the video that is on the stream does give a great representation of how this protein pump actually moves and changes shape to get particles in. So a perfect example of this protein pump, there is a protein pump that's actually named the potassium sodium pump, and that pump actually takes sodium and potassium ions into and out of the cell in order to create equilibrium for the cell. Okay, so our next two types of active transport are exocytosis and endocytosis. I'm not sure why this just says exocytosis. So let's start with endocytosis. I got a picture for you in a second here. But if we're talking about endocytosis, what happens is the cell membrane is going to infold or fold inward and take in a bulk amount of particles. So like a whole bunch of particles. These particles could be solids or these particles could be liquids, either one. But think about going to Sam's or Costco and you load up a whole bunch of stuff in the cart and you pay for it and then get it out the door. Same thing here. You're talking about big, bulky particles and a lot of them. So the cell membrane is going to fold inward around the particles. The cell membrane is going to make a vesicle around the molecules or particles and bring them into the cell. A lot of times this will happen with larger carbohydrate molecules, and then when there has to be a whole bunch of them that has to come in. Endo means enter, exo means exit. So exocytosis is when you've got big, bulky substances, hormones, wastes that have to exit the cell kind of in a mass amount. Again, like you're going to Sam's or Costco. So what happens is the vesicle, whatever substance, solid or liquid, a vesicle is going to be full of the solid or liquid particles. It is going to move to the cell membrane. It is going to fuse onto the cell membrane. That cell membrane is flexible, remember? So the cell membrane is going to change shape. It's going to open up the cell membrane and it's going to expel all the contents outside of the cell. Again, some examples would be hormones or wastes released from the cell. Like hydrogen peroxide would be a waste for cells. So I'm not going to make you draw this, but let me just give you a visual here. Endocytosis, you've got all of these particles. They are gathered, okay, into the cell membrane. The cell membrane enfolds all of these particles, whether they're solids or liquids, get inside of that little cell membrane enfold, okay? And then that cell membrane starts to pinch off a little bit, and then it pinches off more, and then finally you have a vesicle that's formed around whatever particles are coming in. If these particles are solid, we call this phagocytosis or cell eating. If these particles are liquids, we call it penocytosis, which means cell drinking. If substances are leaving the cell, that's exocytosis, again, exo exiting, forces materials out of the cell in large amounts or very bulky, large substances. So again, you've got these substances, could be liquids or solids, that are engulfed or enveloped into this vesicle. The vesicle moves along, it gets close to the cell membrane, fuses to the cell membrane. The cell membrane moves a little bit, separates more, and then those substances leave the cell. Energy has to be used by the cell in order to do this. So this is considered active transport. Okay, you guys, you know what you're supposed to do. I will see you tomorrow, and thanks for being patient.